Dear students, the topic of this lecture is the skin. We included the skin in the third semester to the so-called neuroanatomy and sensory organs, because the skin can be regarded as a sensory organ. However, the receptors which are found in the skin will be discussed later in another lecture. The surface area of the skin is approximately 2 square meters, and we have two types of skin. We have a thin skin, which is hairy skin, so it contains hair follicles, and we have the two most frequently found glands, the sweat and the sebaceous glands we studied in the first semester. This covers the majority of the body. And we have a so-called thick skin or glabrous skin, which doesn't have a hair follicles, so this is a kind of hairless skin, and only sweat glands are found. This is typical for the palm and the sole, and also on the fingertip. The layers of the skin, we studied the epidermis in the first semester, which is the stratified keratinized squamous epithelium with no vessels, as we studied in general in the epithelium. And the thickness may vary according to the type of the skin. In the thin skin, not more than 100 micrometer in thickness and 25 cell layers max. And in the thick skin, we have 10 times thicker thickness, so approximately 1 millimeter, and the number of the cell layers also higher, can be up to 100. The underlying connective tissue, called dermis, has a double role at least with the connective tissue papillae. It provides better mechanical support for the epidermis, and it supplies the skin, so the blood supply is also provided by the dermis. So the blood supply of the epidermis is provided by the capillaries found in the dermis. And this is where we see the majority of the glands and the hair follicles. The epidermis and the dermis together are called cutis or skin. I put it in bracket because it may vary in the different histological textbooks. We teach this way, that the epidermis and dermis is the cutis and or skin, but in some textbooks, if you add the subcutis, which is another connective tissue layer, then this will be called skin. In our case, we call this rather integumentum commune. The subcutis is also connective tissue, which contains fat pads. I will mention the skin appendages later, the hair and the nail, and of course we recall the glands as well. The epidermis is well known for you from first semester. The first layer is the basal layer, which has a high mitogenic activity. That's why it's darker than the rest. And we have then the polygonal layer, that is the thickest part of the whole cellular layers. Typical cell type is a prinkle or spiny cell, because of the desmosomes between the adjacent cells. Then the third one is the so-called granular layer, because of the basophilic granules contain keratohyalin. I will return to this later. This is the last cell layer which is still living with nuclei, but then the remaining, the other layers do not have any more nuclei. These are dead cells. In the thick skin, we are able to distinguish a little shiny layer, the stratum lucidum, and then the rest is the stratum corneum. The corneum or cornified layer can be further divided into two parts. One is quite compact and densely attached to the underlying layers, that is the stratum compactum, and the other, which starts to be shed, is the stratum disjunctum. This is not so well seen in this photo. In case of the thin skin, the lucidum is not so well seen, and the cornified layer is much thinner. The first two layers, the basal layer and the polygonal layer, can be found in the non-keratinized version as well, and this is called together as Malpighian layer. The layers, what I mentioned so far, were according to the keratinocytes. This is the major cell type in the epidermis, but we have additional cells such as melanocyte, Langerhans cell, and Merkel cell. Melanocyte is for UV light protection, Langerhans cells for antigen presentation, and Merkel cells is a receptor cell, a mechanoreceptor. We will discuss this later. So keratinocytes, that is the major cell type. According to this, we have the different layers. Basal layer again with mitogenic activity. And then we have the stratum spinosum with these prickle cells. And I told you the desmosomes give this appearance. And inside the cytoplasm, we see tonofilaments attached to the desmosomes. So this way the anchorage is transmitted to the cytoplasm of the cell. Then we have the stratum granulosum with basophilic granules containing keratohyalin. It has two major components. One is the tonofilament and the clumping of the tonofilaments provided by the profilagrin. And it's interesting that from the so-called lamellar bodies of the stratum granulosum cells, we have a release of extracellular lipid, and this will be important for the important sealing against water loss. I will mention it later. The stratum corneum is the uppermost layer, so we are upside down now. And here you see the synthesis of the keratin, which has two components, the tonofilaments transformed into keratin filaments and the profilagrin into filagrin. These two together form the keratin. So in the epidermis, we have a significant synthesis of the keratin from the bottom to the uppermost cells, and at the same time the cell will die, and in the stratum corneum we don't have any more living cells. The cells lost the nuclei, and we have a special envelope related to the cell membranes in the cornified layer. In the inner surface of the cell membrane, we have a special protein called involucrin, and on the outer surface, we have a special lipid called ceramide, and this is linked to the cornified envelope with covalent bond. Epidermal turnover can be interesting in some cases. Normally, it is between 50 to 75 days approximately. But in some cases, we have a much faster turnover and much faster desquamation on the surface, which is typical, for example, for this autoimmune disease called psoriasis, when the turnover is almost just 1 week. This is the polygonal layer with prickle cells, and you see the tiny extensions of the cells where the cells are bound to the adjacent cells with desmosomes. The next cell type in the epidermis, as I mentioned earlier, the Langerhans cell. With scanning electron microscopy, you see a quite nice flower-like cell. This is able to detect the antigens from the surface. That's why this is part of the MPS, mononuclear phagocyte system or macrophage system, and these cells will present the antigen to the lymph nodes, for example. So this cell type will be a part of the immune system. We have melanocytes with the regular hematoxylinosin staining. These are quite pale cells, so we are not able to see them in the slides. But if we use special immunohistochemistry and antibodies with immunohistochemistry, then we are able to visualize these cells. The cells have several processes, dendrite-like processes, and that's why the cells are able to transfer the so-called melanosomes to the adjacent keratinocytes. This type of secretion is called cytokine secretion, which is stimulated by the UV light or special factors such as ACTH, adrenocorticotropin, or MSH, melanocyte stimulating hormone. We study these hormones in the in this semester later. Some diseases are related to this. For example, the Adizone disease, when we have significant pigmentation in the skin and the oral mucosa, for example. Tumor necrosis factor alpha also able to stimulate this process. The major content of the melanosome is the melanin, which is synthesized from tyrosine. You studied this in biochemistry. The key enzyme is tyrosinase, and if we have a mutation of this enzyme, this leads to a special phenomenon called albinism. Otherwise, in the darker skin, we have more larger and more superficial melanosomes, so this causes the darker skin in general. We have two types of melanin. We have eumelanin, which provides the brown-black color, and the pheomelanin, which gives the red-yellow color, and the combination of these will result in a quite high variation of the skin and later hair colors. I put here another possibility to darken the skin, but it's not a real mechanism, the sunless bronze. In this case, the dihydroxyacetone is bound to the proteins in the stratum corneum only, so that's why with shedding of the uppermost cells, this chemical will be removed also, so doesn't provide a protection against UV light, just a temporary darker skin appearance. The fourth cell is a receptor cell. That's why, for example, one reason why the skin is regarded as a receptor organ, but this cell is only in the epidermis, and we have lots of other receptors which will be discussed later in the skin, but these are located in the dermis or even in the subcutis. The Merkel cells are mechanoreceptors located in the epidermis, so between the keratinocytes. It will be discussed later. This is a very important topic, the important molecular anchorage between the epidermis and the dermis. So this shows the basal layer and then the underlying connective tissue, where we have the junction between epithelium and connective tissue. Usually we have basement membrane. And this is what you see here first, the major layers of the basement membrane. First of all, the darker violet and the lighter violet together is the basal lamina, having first the lamina lucida or rara, this darker violet, and then the lighter is the lamina densa. These two together again the basal lamina, and if we add the dark blue, the fibroretical lamina, these three together will be the basement membrane. So basal lamina, the two violet, and the fibroretical lamina together is the basement membrane. The anchorage provides or includes several couplings. The first is the so-called hemidesmosome, which couples the basal layer cells to the lamina densa, the second layer of the basal lamina. The lamina densa contains collagen type 4 and perlecan, and these yellowish anchoring filaments, what you see here, are those couplings which are able to couple the basal part of the basal cell layer to the lamina densa. These are usually laminin or collagen type 17, these tiny light yellow structures. Then we have the next bridge from the lamina densa to the collagen fibers, light green, and these are a bit thicker elements anchoring fibrils, type 7 collagen usually. And then we have a third bridge which transmits the force from the collagen fiber to the elastic fiber. These are provided by the microfibrils. So this way we have three steps, three bridges between the basal cell layer and the underlying connective tissue fibers. In some cases, unfortunately, autoantibodies are produced in our body against one of these elements. These can be a very serious autoimmune disease. In a minor case, only blisters are formed, but in a more serious case, even the whole epidermis can be removed, called epidermolysis. In the dermis, we are able to distinguish two layers. This is again very important question in histology. The upper layer, which contains loose connective tissue, this is the papillary layer because it has also the connective tissue papillae. And in the papillae, we have capillary loops, so this is from where the epidermis receives the blood supply. Also, we may find receptors such as Meissner's corpuscles you study later. This loose connective tissue contains type 1 and type 3 collagen fibers, and these are fine collagen fibers. In the second layer, which is the stratum reticulare, we have dense connective tissue with much thicker collagen fibers, type 1, and elastic fibers. And in some cases, we have smooth muscle cells also in this region, and this is called tunica dartos. We studied in the genitalia because it was typical for the penis and the scrotum. So this is the stratum reticulare where we can find them, or reticular layer. In the reticular layer, the collagen fibers are arranged in rhomboids, and the long axis of the rhomboids called Langer's lines. Usually, the Langer's lines coincide with the so-called relaxed skin extension lines or tangent lines, Kressel lines, and this is what you have to regard when you do plastic surgery. You have to excise parallel with these relaxed skin tangent lines because then you have better healing and faster healing of the wound. From outside, you see the dermal papillae in ridges. These are the so-called friction ridges, which provide an individual fingerprint, dermatoglyphics, and this can be used for identification. And another line, for example, at the level of the joints called flexure lines. In this case, the skin is coupled to the underlying joint capsule, and that's why we have these invaginations, basically. Under the skin, so-called skin, we have the third layer, the superficial fascia, or called subcutis or hypodermis, which contains significant fat pads called panniculus adiposus. This is important for energy store, thermal insulation, and also is kind of shock absorber against mechanic stress. And between these fat pads, we have collagen septae, retinaculum cutis, dense collagen fibers, and it couples the subcutis to the underlying skeletal elements, periosteum, for example. That's why it's make the skin stable. This is important, especially in those areas where we have vertical connective tissue fibers in the sole, in the palm, to be able to stand, in stable position or to grasp object, and the skin is not mobile. But in other cases, other areas such as the eyelid or the foreskin, we need mobile skin. So that's why we have almost no subcutis in this region. The glands, it's important recapitulation at the same time. We studied, 3 glands in the skin. So it was a basic rule that if you see a stratified keratinized squamous epithelium, then only these 3 glands are the options. So no mucous and serous glands, that is a big mistake. Only these 3 glands are the options. The most frequent would be these 2, the sebaceous gland and the sweat gland, but in some areas we have a third type, the so-called odoriferous gland. Sebaceous glands are branched alveolar glands with holocrine secretion, and usually they open into hair follicles, which serve as a duct. And in some areas we don't have hair follicles, but we have still sebaceous glands. I listed them here. It is, for example, in the lips or inner surface of the prepuce and minor labia, glans penis or glans of clitoris, and in the areola of the breast around the nipple. And these glands are also missing in the thick skin, which covers, the sole and the palm. The odoriferous glands are simple coiled tubular glands. That's why we see many cross sections. And as we see in darker color, the duct of the secretory portions. They operate with merocrine secretion. That is the most general, secretory mechanism. We studied this, and they open on the surface. But in some cases, these glands are missing. Again, these are basically those areas where we have sebaceous glands with no hair follicles, so lips, inner surface of the prepuce, minor labium, glans of the penis or clitoris. And the innervation is sympathetic, but in this case we have a very unique neurotransmitter. This is not the norepinephrine, which is general for the sympathetic nervous system. Here we have acetylcholine, so that's why it's cholinergic sympathetic innervation. The third, the odoriferous gland with apocrine secretion. Typically we have large lumen and or irregular surface because the cells are not in the same phase. And AAA is the typical area or areas where these glands are located: axillary, areolar, anal region, plus the genital regions as well. In some cases, in the skin we see other apocrine glands with different names. For example, the ceruminous glands in the external acoustic meatus, producing the creamy cerumen usually, and we have some other glands, apocrine glands, in the skin of the eyelids, the glands of mole or ciliary glands. They are under the regular adrenergic sympathetic innervation. The blood supply of the skin, as I told you, the epidermis receives the blood supply from the dermis, where we have two arterial plexuses. We have first a deeper plexus, arterial plexus, also called subdermal plexus, and we have another plexus under the papillae, connective tissue papillae, called subpapillary or superficial arterial plexus. These are connected by quite straight vessels to each other. And from the subpapillary plexus, we have capillary loop to the connective tissue papillae, from where we have then the venous drainage backward. And we have basically the same plexuses. So one is the superficial or subpapillary venous plexus, the other is the subdermal or deep. But in case of the veins, we have a third one, the middermal venous plexus as well. They receive otherwise the blood supply from the underlying subcutis, what the next figure will show. But before I go there, I like to highlight something that in some cases, in those areas which are quite external, such as the fingertip or nose, ear, where we have more risk for heat loss, we have shorter shunts or arteriovenous anastomosis, so we don't have so complicated capillary system to slow down the circulation and to have more heat loss. We have a protection against this. That's why we have a shorter variation between the arteries and the venous. Now this is mostly for plastic surgery, showing the skin and together with this the subcutis. We have two options. We have a direct cutaneous branch of the arteries which supplies the subcutis and then the dermis, and in the dermis those plexuses what I mentioned. But we have another option. We have a deeper artery which supplies some skeletal muscles first and then the skin above, or the fascia and then the skin above. So you have to regard these musculocutaneous or fasciocutaneous arteries if you do plastic surgery. The innervation of the skin is also interesting because we have nice pattern of the skin innervation, and each spinal nerve has a corresponding skin area called dermatome, which seems to be quite irregular, but if we are our four extremities, then these patterns, these stripes of the skin, so the dermatomes will be perpendicular to the longitudinal axis of the body, to the longitudinal axis of the spinal cord, and then everything becomes logical. We have a significant overlap between the adjacent dermatomes, innervations. That's why very rare when we have a sensory loss in one dermatome, because it may receive innervation from the adjacent dermatomes as well. The functions of the skin. It is receptor organ, and that's why we put the skin into this semester, but the receptors will be discussed later. And it has significant mechanical and chemical protection. I mentioned already the desmosomes, the anchorage system earlier between the dermis and the epidermis. It has an important role in the thermoregulation with passive and active elements. Passive includes the fat pads in the subcutis, active the sweat gland mechanism, sweatings, and the vessels, and the arrector pili, which is a tiny smooth muscle. And we have photoprotection related to the skin with two components. One is the Stratum Corneum, which is able to reflect the UV rays, and the melanin itself, I told you earlier, in the melanocytes, which is a scavenger, which is able to bind the harmful free radicals. And the permeability barrier was also mentioned briefly, because I told you the extracellular lipids from the Stratum Granulosum, granular layer, together with the other elements in the Stratum Corneum, they provided important sealing against water loss, and that's why it made it possible in the evolution that the reptiles were able to leave the ocean and become, they were able to use terrestrial life or live in the terrestrial life. Protection against pathogens, also important. On the skin we have a negative pH because of the sweat and the fatty acids. We have also RNases, so we are able to break down the RNA, which is important for laboratory works in case of molecular biology. But now the kits are much more stable, so we don't need to pay attention too much on this. We are able to synthesize several factors, vitamin D, for example, which is important, especially now, against coronavirus, it seems. Other cytokines and growth factors, such as the epidermal growth factor. It's important in the tumorigenesis. And I read a recent paper which explains why in special populations, typical for the Koreans, for example, East Asia, who have a special allele in this gene, ABCC11 gene, double adenine instead of guanine, and that's why they don't express a special pump which would be important for some materials in case of the odoriferous glands to be secreted on the surface, and these are missing, and that's why the bacteria don't break down these special substances because of the lack of the pump, and they don't have basically body odor. They don't use deodorants, and they have also very special cerumen in the external acoustic meatus. They don't have creamy earwax, they have a very dry, odorless earwax. I asked the South Koreans, and it's true. So, another important thing, consideration regarding the permeability barrier, the lipid layer, what I mentioned earlier. If we want to apply special treatment on the skin, and which are hydrophilic, then we have to somehow make them lipophilic to be able to pass this barrier. That's why these are usually included in liposomes. So in liposomes which are able to pass the lipid layer. So tattoos, I don't want to show the other variations. I just put here because it was quite recent, sad story. Kobe Bryant air crash, and LeBron, his friend in Los Angeles Lakers, made a tattoo on the honor of him. And the tattoo in general is made by special needles under electric current, and the ink is introduced into the dermis layer. This is what I wanted to tell. Now the hair follicles. In those areas where we have hair, so not in the thick skin, palm and sole, and some other regions such as the lip, nipple, and the glans penis or clitoris, and the inner aspect of the foreskin and a major labium and minor labium, where we don't have skin. In the other area, we don't have hair follicles, sorry. We have otherwise hair in the other regions. And here we have to mention the three parts of the hair follicle. The lower part is the inferior segment. Then we have a narrow point, the isthmus, and the uppermost part, again enlarged. This is called infundibule. The inferior segment contains an enlargement at the bottom called bulb, and you see a quite pale invagination. This is dermal papilla, which provides the blood supply of the germinative matrix around. With higher magnification we are able to see it later. And you see in a special angle a tiny smooth muscle, the arrector pili muscle, where it joins to the hair follicle, and they form together with the hair follicle an angle where you see the majority of the sebaceous glands. This unit is called pilosebaceous unit, and with the contraction of the smooth muscle, which is also under the influence of the sympathetic nervous system, it acts on the secretion of the sebaceous glands, compressing the glands. The lowermost part of the hair follicle contains the bulb, and we have the dermal papilla from below with the capillary loop for the blood supply of the germinative matrix. This is the germinative matrix. So basically similar to the epidermis. Here we have the cells with mitogenic activity, and they multiply, they divide, and they create cells shifting upward in the medulla, lighter yellow, and in the cortex, a bit darker yellow. You see here also melanocytes between the keratinocytes, and they will provide the color of the hair. The third component or layer of the hair is the darkest yellow, that is the cuticle. So the hair has three layers: the medulla, cortex, and the cuticle. All of them are formed from the underlying germinative matrix, the transformation of the living cells into keratin, basically. And we have then the inner root sheath with pink. The middle pink is the innermost pink is the cuticle of the inner hair cell, inner root sheath. The lightest pink is the Hux layer, and the darkest pink is the Henle's layer. So these three would be the inner root sheath. Then we have the outer root sheath, which basically corresponds to the lower two layers of the epidermis, so the polygonal layer and the basal layer. Then we have the glossy membrane, which is regarded as the basement membrane of the epithelium, and then we have a connective tissue sheet around. It lists the several types of the keratin, not necessary to know, but we have different keratin as it was in the epidermis. This is from our regular slide. This is the bulb, the thickest part of the hair follicle, including the germinative matrix in this arch around this paler dermal papilla, where we have the blood supply of these cells, and you see very dark cells, melanocytes, located together with the dividing cells. And then in the middle, number 3 is the medulla, number 4 around, a little bit darker, is the cortex of the hair, and the fifth layer would be, number 5 is the third layer of the hair called a cuticle. And the number 5, the cuticle of the hair, with interdigitations, is connected to number 6, which is the innermost layer of the hair follicle, inner root sheath, that is the cuticle of the inner root sheath. These are connected to each other. You see some connection still here, but otherwise these are detached already from each other. And number 6, otherwise altogether, the inner root sheath, which has 3 layers I mentioned earlier. And then number 7 is the outer root sheath, which corresponds to the stratum basale and polygonale. Number 8 is the basement membrane of the epidermis-like cells, so the epithelial cells, that is the glossy membrane this way, and number 9 is a tiny connective tissue sheet around. This is what I wanted to show you, how the cuticle of the hair is seen as the tile of the roof upside down, and the interdigitations would be together with the cuticle of the inner root sheet. In cross-section, you see the hair inside, and we are not able to distinguish between the medulla and cortex, but here you see in the outer part that the cuticle of the hair is still connected to the cuticle of the inner root sheet, but in this area it was already detached. In general, the more intense pink is the inner root sheet. You don't need to distinguish the outer Henle. The second is the Huxley and the innermost cuticle of the inner root sheet. Just inner root sheet, it's enough to say for the pink, intense pink. Then the rest will be the outer root sheet with the basal layer and the polygonal layer originally from the surface. Then the glossy membrane, which is the basement membrane, and then we have the connective tissue sheet around. The growth of the hair has a special cycle. This is what you see. The first and the longest cycle is the so-called anagen phase. The longest phase of the hair cycle is the anagen phase, which lasts for years, 2 to 6 years. This is when the germinative matrix cells multiply, and the hair is created and shifted upward, and we have a synthesis of keratin at the same time that of the cells upward. And then we have a so-called catechin phase, the transition phase, which lasts only a few weeks, when the dermal papilla starts to be separated from the bulb, and the hair shows a special club in the end. Then it continues in the telogen phase, the resting phase, which lasts for 1 to 2 months, let's say. Then we don't have any growth, and the dermal papilla is completely separated from the follicle, so that's why it doesn't have blood supply. And then returns to the anagen phase again, when the dermal papilla unites with the bulb, and then the germinative matrix starts to multiply again, and the new hair will push out the previous hair. This is the shedding mechanism. I put here some data. The normal hair loss is between 50 to 100 days, so we have a normal loss as well. If it's synchronized, which is typical for the animals, it's called shedding, but in human it can be present also in case of newborn. The chemotherapy is harmful for the germinative matrix and also the blood supply. That's why we have hair loss, but thanks God it's a reversible process, so after the chemotherapy the germinative matrix and the blood supply will be repaired and new hair can be growth. And the cross-section, it's interesting. If somebody has straight hair, we have round. If we have curly hair, we have oval cross-section. And the effects of the hormones, the androgens, induces the hair loss called alopecia because of the shorter anagen phase. And the color of the hair, I told you, depends on the amount of type of the pigments, eumelanin and pheomelanin. This is a movie showing the importance of the hair, make it quite, interesting. And some practical considerations, what I don't want to detail. And at the end, I like to tell something about the nails. The nail is also a keratin, structure, nail plate, produced by the matrix here in this angle, having different parts, dorsal, middle, and the ventral part. You don't need to know this. And then the rest is called a nail bed. So the matrix is this angle, and then the nail bed is the distal part. and this would be here the nail plate. The lunula, what we see from above, is the end of the matrix. The green is the matrix in this case, and lunula corresponds to the end of the matrix, and the rest is the nail bed, basically. So this is not correct from this aspect. It focuses on the different parts of the matrix, but this part should be already the bed, as this shows. And we have two protections, coverings, called eponychium, between the proximal part of the skin and the nail plate, and we have the hyponychium between the nail plate and the fingertip. Well, the other important thing is that the matrix, labeled in green, and the nail bed, labeled in yellow, area is special because here we don't have subcutis. So here the connective tissue under the matrix and the bed is tightly connected with collagen fibers to the periosteum of the skeletal elements. So if we have a bleeding here because, for example, with hammer accidentally we hit our fingertip and the blood will be produced in this region, it's very painful because of the very tight connection between these two structures, and that's why we may need to incise the nail plate to release the blood. This way we can relieve the pain. It's a new data, it's a quite popular topic now, the stem cell induction. In this case, pluripotent stem cells were induced to create hair follicles, and there was an organoid, there's a special in vitro skin precursor, let's say, and organoid in a sphere. And from this pluripotent stem cells with special factors, researchers were able to induce the formation of hair follicles. This is what you see. So it's a topic of the research recently. And at the end, I wanted to show some extreme, nail plates, and at the same time, I would like to thank your attention.